Thank you for the invitation. It's a new field for me. I am a B-cell biologist and scientist working with B-cell diseases. And a couple of years ago, I read the preliminary reports, first reports of Dr. Fluga, and got really interested and invited Jonas Blomberg to come to Linköping, where I work, to sort out what is chronic fatigue syndrome. So most of my background is autoantibodies, Epstein-Barr virus, trying to understand the very basic biology of B cells. Now, I will give a background to a couple of papers that came, one of Navio a couple of months ago, and now one paper last week, both dealing with metabolomics, so ämnesomsättning in Swedish. This seems like we are right now in a breakthrough where there are accumulating evidence for a chemical signature, or chemical fingerprints. One fingerprint in the Navio paper in Proceedings of National Academy of Science, and another in the Yamano paper in the Science Report just last week. I will speak a little bit, give you a background, and Jonas will explain the results to you after me. Now we can see that it's a very complex situation right now because we have chronic fatigue syndrome really affects mitochondrial energy metabolism, autonomic nervous system, immune functions, gastrointestinal functions, microbiome, and neuroendocrine system. And to find diagnostic tools or chemical signature for all this system is not easy. So some things might be complicated. At least for me, it was really complicated because I started only a couple of months ago to read about the disease. The background, and I see it as an outsider's attempt. I'm the outsider. How we should interpret these metabolic changes. Navio speaks about cell danger response, acute versus chronic. I will tell a little bit about that. And metabolic changes, it seems to be a low energy level, low metabolic state. And I might agree with Øystein that the low metabolic situation could be an attempt to produce ATP from several sources. We must understand also that mitochondria are really not only ATP chemical energy producing organelles, but also signaling organelles. And they are critical for immune response. It's actually a new field, immunometabolism, that is born. Cell danger response is a very evolutionary old response to cope with various threats. Chemical, like metals, plasticizer, chemical flame retardants. Physical, like heat, salt, pH balance, radiation. And then biological, where my interest is microbes, virus. But also psychological trauma can activate CDR. CDR stands for cell danger response, to produce chronic inflammation. We find this response down through the lower species in model systems like C. elegans. And it focuses on a limited number of metabolic pathways that are utilized to respond to these attacks or threats. It's coordinated in the brain via chemosensory integration and whole body microbiome metabolism. here is a picture that is impossible to see, but the slides will be available to anyone interested. What Navio puts forward is that 8 points, I will go through some of them, but there is a shift, cellular shift from, in metabolism from polymer to monomer state. And there is a shift in the membrane that stiffens to sort of prevent microbes from entering, but there is also from the mitochondria produced what we call free radicals or reactive oxygen species, heat shock proteins, and antimicrobial proteins of various type. And what we have found is also mitochondrial DNA that comes out as a little signaling molecule. We have release of eicosanoid, leukotrienes, ATP, and cytokines. It's like interleukin-1, TNF, and so on, that affects dendritic cells and T cells. But it also reaches a central nervous system. There are receptors for interleukins and TNF in the hypothalamus. Sorry. These, for instance, interleukin-1, I'm not here. Interleukin-1 reaching the hypothalamus triggers a sickness behavior and low fever, fever centra. Here are examples in this article, or the previous article of Navio, of several metabolites that have upward or downward, here again 8 others, upward or downward metabolic pathways. And he describes this as healthy development upwards and sort of winter maintenance, where we historically had low calorie intake in the winter, which is opposed to summer, where we had affluent food and we could sort of collect energy reserves. And this is also characteristic for inflammatory state. The paper in PNAS a couple of months ago describes similarities between chronic fatigue syndrome and a hypometabolic state that are seen in throughout the evolution in species like C. elegans. And he describes a condition called Dauer. It's a German word meaning long-lived or persistence. In Swedish, långlivad, uthärdande tillstånd. When energy reserves are limited, it's not like hibernation, because they're also animals that go to hibernation, or ide, as we say, som igelkottar och björnar. They have also lowered temperature, but this is not seen in CFS patients. He describes and finds out of over 600 metabolites, 8 for men and 13 for women, that are differently expressed and actually decreased in this case. There are 20 that are differently expressed, but 8 are decreased, and this is very similar to this hypometabolic condition, Dauer. But it's opposite to acute cell danger response and opposite to metabolic syndrome. This is like a very clear signature, and he speculates a little bit that when, let's say, an infection comes and it cannot be handled properly by the acute cell danger response, then the program changes into a low metabolic state that has a sort of a function to to starve and to and avoid the microbes to take over. So it's like starving the microbes. And one of the top candidates are sphingolipids and glycosphingolipids and phospholipids. That explains most of the differences in this chemical profile. Sphingolipids are highly conserved components of the cell membrane and form so-called lipid rafts. They're domains for receptors and cell signaling, regulate cell division, maturation, and terminal differentiation. But sphingolipids are also important in viral infection. Several viruses exploit membrane components, such as sphingolipids. It's important for viral entry. You see names here like HIV, rhinovirus, measles, Ebola, Simbid. And for assembly and budding, we have hepatitis C and HIV. Viruses manipulate also cellular signaling to their advantage, like flu. And my field here with that I'm a little bit more familiar with this Epstein-Barr virus modulates these lipid raft microdomains and vimentin cytoskeleton for signal transduction and transformation. It's an excellent overview of the situation where we have metabolics meeting the immune system, written by Weinberg and et al. In Immunity, that I can recommend. Very nice pictures. And we see here, they call it a signaling platform, the mitochondria that we see here. It gives signals in redox. This is the hydrogen peroxide and free radicals. And as Dr. Fluge explained here, the lactate production from the TCA cycle is important. And here we see also that it's important for protein modification, acetylation, and fatty acid synthesis, and ketoglutarate is signaling also. So it receives and sends out signals, this little organelle in the cell. And it activates immune responses. This pathogen-associated molecular patterns and danger-associated molecular pattern signal down to activation of another organelle called inflammasome. And here, mitochondria are highly active in contributing to mitochondrial antiviral proteins that assemble here together with calcium, cardiolipin, and hydrogen peroxide that reduce. And then proforms of cytokines are cleaved into interleukin-1 that are functioning. And this is very important concept to see the involvement of mitochondria here. But it also generates formyl peptides and mitochondrial DNA that DNA sensors are reacting to and activating other pro-inflammatory genes like interleukin-6 and TNF-alpha. Our own studies, this is so from our group in Linköping, we find a highly interesting phenomenon, we think, that has not been reported before, that B lymphocytes are, we call it webcasting. When we activate B cells, particularly innate B cells, B1-like cells, with viral peptides, viral nucleotides, they are triggered to release mitochondrial DNA nets. Here we see example of these nets, and these are DNA molecules that we see here, stained with DNA stainers. it's broken down by DNase enzyme, for instance. It's known that these mitochondrial DNA, when they come out, they activate monocytes, microglia cell that we heard about before, to produce interleukin-1. They come out into the circulation, and we can measure this now with a PCR technique. And I wonder how the situation is in CFS, of course. It's just beginning. So, as a summary, we see that cell danger response can be acute and chronic, and these are quite different in their metabolic profile. And what we see in CSF is a hypometabolic state that is different from acute infection. And mitochondria are signaling to the nucleus and circulation and collaborate with the immune system. And my own sort of very simple hypothesis here, our synthesis, is that the initial agents like virus, bacteria, trigger innate B cells to produce antibodies. These B cells are then continuously fueled at a low level by altered self-antigens, antigenic mimicry, to produce antibodies that react with self-structures. We have examples of heat shock protein, myelin, oxidized low-density lipoprotein. And these activated B cells, regulatory B cells, release mitochondrial inflammatory signals and antibodies, we should say. So I completely agree with you that antibodies could be very important here also, together with mitochondrial DNA. This affects the whole system into a metabolic, hypometabolic state. And of course here, with the central position of B cell, it's so interesting to see the effect of rituximab. And I would be delighted to see also effects of anti-interleukin-1 that we discussed before. There are anti-interleukin-1 receptor pharmacological agents around. Thank you. And now I hand over to Jonas.
Thank you.